This is a production of Cornell University.
Thanks for the introduction, Chris. I also grew up in Ithaca and was a research assistant at Cornell back in the summer of 2008. So it's been a real pleasure to be back here again, even if just over Zoom. After my time at Cornell, I went off to grad school at the University of Michigan, as Chris said. And while I was there, I studied range expansion dynamics of plants in response to climate change. Specifically, I focused on biotic interactions between range expanding plants and herbivores and pathogens. The core of my work was a series of transplant experiments across a latitudinal gradient, and in these experiments, I quantified how biotic and abiotic processes affected plant growth and survival. Data from these field experiments gave me a chance to study how these processes varied across spatial scales ranging from meters to hundreds of kilometers, and to quantify how these processes affected plant populations. I also investigated the effects of various global change drivers on plant populations using meta analysis. Ultimately, this work was motivated by the desire to both understand how climate change will affect these forests and the hope that this knowledge would inform management practices. These are my research themes: creating models of ecological processes for individual plants, scaling to populations and landscapes, and using this information to inform management. However, it's hard to manage processes that are occurring on timescales of centuries, and I decided I wanted to work on a plant ecology topic that had more immediate relevance to people's lives. So, after my PhD, I began to apply these same research themes to a new topic: airborne pollen. In my talk today, I'm going to start by covering the current state of affairs in airborne pollen, and then move on to describe the spatio-temporal models of airborne pollen that I've created by quantifying pollen production, plant location, and flowering phenology. Through the talk, I'm going to show you how I've applied these same methods to urban trees and ragweed plants in Detroit, to juniper trees in Texas, and then I'm going to tell you a little bit about my future research plans of applying similar methods to trees at continental scales. Almost all gymnosperms and at least 10% of angiosperms are wind pollinated. In temperate regions, like Ithaca, this includes most trees and lots of important crops, too. For these plants, understanding airborne pollen is critical for understanding gene flow, and this is relevant to research that's being done now at SIPS. For example, pollination is required for hemp crops that are being grown for grain, whereas airborne pollen can interfere with hemp being grown for CBD. Another relevant agricultural issue is the spread of herbicide resistance genes across landscapes. Similarly, understanding airborne pollen is important for paleoecologists that reconstruct plant communities using pollen grains in sediment cores or fossils. Pollen also directly affects human health by triggering allergic reactions, and an understanding of airborne pollen is important in all of these examples and to many examples that I haven't listed. Over the next few days, I'm looking forward to talking with many of you about this topic and how my research themes might apply to your study systems too. That being said, for most of today, I'm going to focus on airborne pollen through the lens of allergies and human health. Pollen allergies are becoming increasingly common around the world, and rising carbon dioxide concentrations also cause plants to produce more pollen and pollen that is more allergenic. And even though pollen allergies are sometimes dismissed as just a nuisance, they make life miserable for millions of Americans and people around the world. They also have big societal consequences, including millions of lost school and work days every year, and can also trigger potentially fatal asthma attacks. Most of what we know about airborne pollen comes from pollen monitoring stations. There are about 80 of these stations in the United States, which are run through the National Allergy Bureau network, and each of these stations takes integrated daily measurements. Here's a map of these monitoring stations. If we zoom into the northeast, you can see that the nearest station to Ithaca is in Rochester, about 75 miles away. The current paradigm in this field is to just use data from the nearest monitoring station. In other words, it is standard practice to assume that pollen concentrations in Ithaca are the same as they are on one roof in Rochester. In fact, when you see pollen concentrations reported in the media, what you're usually getting is yesterday's measurements, likely from tens of miles away from you. Also, while the forecasting methods that are used by companies are usually proprietary, one of the poorly kept secrets is that a lot of these forecasts from leading websites are actually based off of allergy medication sales, which is not always the best proxy for airborne pollen concentrations. So, even in the best case, these predictions of airborne pollen generally assume that pollen is homogeneous within regions. Surprisingly, there's very little data on how pollen concentrations vary within regions. One potential explanation for why we don't know more is that most plant ecologists don't know much about allergies, and most allergists don't know much about plants. So I think this has kind of fallen into gaps between fields a little bit. However, there's another really big reason why we don't know more, and that is the methods that are used for sampling pollen. And traditionally, two types of samplers have been used, the rotorod and the Burkhardt sampler. These samplers were designed about 70 years ago and haven't changed much since then. They're bulky, they need to be plugged into AC power, and they also aren't cheap, which makes it difficult to conduct these large simultaneous measurement campaigns that we need to figure out how pollen varies over relatively small spatial scales. One of my contributions here was making an updated design of one of these pollen sampler types myself. And I was lucky enough to get some help from a few engineering grad students on this, and I ended up soldering together a fleet of these samplers. The designs for them are currently in prep for submission to an open source hardware journal. With these samplers, I was able to empirically test whether this large assumption of pollen being homogeneous within a region was true. Next, I'm going to show you some results on that. This is a map of Detroit, the study area for my first postdoc, and this map shows simultaneously collected oak pollen measurements across Detroit. The color of each of these circles indicates airborne pollen on a particular day. So this is just one day as an example. And I'd like to note that the color scale here is logarithmic. And the take-home message from this is that pollen concentrations do vary dramatically over relatively short distances. It also shows why using a single station as a proxy for an entire region doesn't work all that well. In fact, measurements from that pollen station shown in the upper right-hand corner of this map, they only explain about 6% of the variation in oak pollen across the area across the season. So, and I should also point out that other taxa had similar results, and concentrations on a particular day often varied by 2 or 3 orders of magnitude across the city. To me, this means that we've been making bad assumptions about exposure to airborne pollen because we haven't been thinking about it spatially. I think it's time for a paradigm shift in our understanding of airborne pollen. In my talk today, I'm going to show you the benefits of transitioning from a phenomenological monitoring network to a spatial modeling approach that includes the underlying ecological and biological processes. I think this has the potential to revolutionize our understanding of allergenic pollen exposure. So, in the next section of my talk, I'm going to walk you through my approach to modeling airborne pollen, which requires answering these questions. First, how much pollen does a single plant produce? Second, where are the plants that are releasing pollen? And third, when are these plants flowering and releasing pollen? Now, each of these are basic research questions on their own, but the combination of them leads to applied solutions to real-world problems. And in fact, even though this is research on plants, the health implications here are strong enough that it was competitive for NIH funding, which supported my postdoc at U of M. So let's dive into pollen production. My goal was to be able to estimate an individual tree's pollen production as a function of its size, but no such equations had been developed for North American tree species, and the equations that had been developed for trees elsewhere generally used pretty coarse methods. I set about creating equations for several tree species, but for most of my talk today, I'm going to show you examples from oak trees because they are common and produce lots of allergenic pollen. At this point, I'd also like to acknowledge Victoria Bankowski, who was an undergraduate student who worked with me for 2 years on a couple of projects, including this one. Here's an example of how I estimated pollen production. First, I selected trees of a range of sizes, and then I borrowed a trick from my days of assessing understory light availability and took hemispherical photos of each tree's canopy. These photos made it easy for me to systematically and quantitatively estimate the number of flower clusters per tree. I then took branch samples and counted the number of catkins per cluster, and then counted the number of anthers per catkin, and finally counted the number of pollen grains per anther. I then multiplied all of these together to estimate a tree's pollen production. So the question is, how well can we predict pollen production based on a tree's size? On the x-axis here is a tree's canopy area, and on the y-axis is the number of pollen grains produced in millions. So let's take a look at the results. The dots here are individual trees, and the lines are regressions. The mean is in blue, and the variation, which is based on bootstrapping, is in gray. So for red oak trees, larger trees produced more pollen, and they did so in a predictable fashion. This held true for the other species in the study too, and it turns out that tree size is a very good predictor of pollen production. That being said, I want to take a moment to focus in on something here. I have pollen production in the millions, but it's kind of hard to get a good gut sense of what 100 million pollen grains means. So I want to put those numbers in some context. The largest white mulberry tree that I measured had about 125 million anthers. Here's an animation of a single white mulberry anther releasing pollen. That single anther has about 50,000 pollen grains in it, but it only takes as few as 15 pollen grains per cubic meter of air for the most sensitized people to have an allergic reaction. So imagine being allergic to white mulberry and having hundreds of these trees in your city. The next step in developing a model of airborne pollen was to figure out where each of these trees were. Now, my study area was the city of Detroit, which is big, and at 360 square kilometers, there's no way to measure all of these trees by hand. Luckily, we're living at a time when vast amounts of new remote sensing data are becoming available, as are processing techniques to manage it. And these can be applied to tasks like tree identification. One of these new data sources is a company called NearMap, which takes high-resolution aerial images of most urban areas in the US a few times a year. And this data set had never been used for tree identification. I combined the multi-temporal aerial images from NearMap with a few other data sets, including LiDAR and a street tree database, and used that to identify trees in Detroit. I'm going to show you a brief overview of how I did this analysis, but if you have questions about the details, I'd be happy to discuss it more in the question and answer session after the talk. Okay, so here's an aerial image of one spot in Detroit. You can see trees, houses, a golf course, and now let's overlay that LiDAR data on top of this. LiDAR provides information about the three-dimensional structure of things. And here, the lighter colors, the white, corresponds to taller objects. With this information about the height of each object, we can then use segmentation algorithms to segment these into canopies of individual trees, or at least large pieces of a tree. And that's shown by these yellow polygons here. I then extracted various indices from the aerial imagery and the LiDAR and used that to classify the trees. The color of each polygon here corresponds to the predicted taxon of that tree canopy polygon. This worked out great for several taxa, but others I couldn't tease apart as well. But even with these limitations, these predictions of individual tree identity are incredibly useful. I'd also like to note that we're on the cusp of tree identification with remote sensing being operational, and over the next few years, these data sets are going to become more and more widespread and accessible. Now, I'd like to draw your attention to these green polygons, which are oak trees. And here's where it gets really exciting. Because we know the area of each of these tree canopy polygons, we can plug in those allometric equations of pollen production to predict how much pollen each of these trees is going to produce. And here's estimated pollen production for each oak tree in this frame. Now, because this is done with remote sensing data, though, we can scale this up. So let's zoom out to the whole neighborhood here. And this is the University District in Detroit. It's an area with a lot of oak trees. Now let's zoom out even more to the whole city. The square on this slide is where the previous slide left off. And here we are, a map of oak pollen production for Detroit based on pollen production by individual trees. This is really cool, and you can just see the differences in neighborhoods between oak pollen production. But the next big question is whether nearby pollen production predicts airborne pollen. After all, people have long assumed that because pollen can blow long distances, that it usually does. Let's take a look. The x-axis of this figure is predicted pollen production around a sampling site, and the y-axis is the average concentration of airborne pollen during the springtime based on repeated sampling. So, to answer the question of whether there's more airborne pollen near source plants, the answer is yes, there's definitely a strong relationship here. This is an exciting finding because it provides very strong evidence that local plants are driving airborne pollen concentrations. In other words, this shows that if you live in a neighborhood with more oak trees, you're likely going to be exposed to much more oak pollen. I'd also suggest that if you're allergic to oak trees, you shouldn't be planting one in your front yard. Now, this is common sense, but we haven't had a scientific basis to justify that before. And so this finding is really relevant for large landowners like municipal governments and points to the possibility of being able to intentionally create lower allergen landscapes. So my major conclusion here is that we should be including allergenic pollen production as one criterion in our tree selection decisions, because the choices that we're making about what trees we plant affects pollen exposure for the people that live nearby. Now, one concern is that this finding could be interpreted as an argument for having fewer trees in cities. I have two responses to this. First, one of the best management options is to just plant different trees. And specifically, we could plant cultivars that produce less pollen. The other response that I have is that allergenic pollen, it's just one of the many ways in which we're interacting with trees. And in almost all circumstances, the ecosystem services provided by trees will dramatically outweigh the ecosystem disservices of allergenic pollen. I'd also like to point out that this same framework that I'm using, understanding processes for individual plants, scaling them up, and using that information to inform management, we can also apply this to some of those ecosystem services that these trees are providing. So I'm going to take you on a brief detour and show you shade as an example of this. So take this tree here in Austin, and then look at it from above, outlined in blue. To understand the shade provided by this tree, we can create a mechanistic model that takes into account the height of that tree and the angle of the sun on any particular day of the year. Here's an animation of modeled shade throughout the day. You can see the trajectory of the sun across the sky and see how these trees shade different buildings. And again, since this is done with remote sensing data, it gives us a way to scale from individual plants to landscapes. And I have to say, having just lived through my first Austin summer, I have a deep appreciation for the shade these trees provide. But let's get back to pollen in Detroit. So we've now covered plant location and pollen production. So let's talk about the other process that we need to predict pollen emissions, which is the timing of flowering. We know that temperature determines flowering time, and we also know that urban heat islands and temperature gradients in cities are common. I therefore hypothesize that there would also be differences in flowering time within Detroit. To assess the relationships between temperature, flowering time, and airborne pollen, I conducted a field study in Detroit where I measured flowering time for a bunch of trees in the city. And I measured the flowering phenology of over 100 trees in 9 different neighborhoods, and those neighborhoods are shown with the hollow symbols on this map here. I then compared flowering time with temperature during the early spring, specifically land surface temperature, which comes from and to get that land surface temperature, I used data from the MODIS sensor on the Terra and Aqua satellites. Here are the results for whether temperature explains flowering. Temperature is on the x-axis, and the timing of flowering is on the y-axis. Each of these small symbols is a single tree, and the large symbols are site averages. And as you can see, the association is very strong, and warmer places flower earlier. We can then combine our knowledge of this relationship with remote sensing-based measurements of temperature to predict the timing of flowering across the entire metro area. So next, I'm going to show you an animation of predicted flowering dates for oak trees in Detroit. Each of these frames is a day, and flowering is indicated by the yellower colors. So you can see that wave of flowering moving from the south to cooler areas in the north. Then, by the middle of May, you can see trees have basically finished flowering. I've now shown you data on the various processes that determine airborne pollen concentrations, including how much pollen is released, where pollen is released from, and when that pollen is released. We can then combine all of these different processes to predict pollen release over both time and space. And next, I'm going to show you an animation that puts all of this together. And this is predicted pollen release for oak trees in Detroit in the spring of 2017. And here you can see the interplay of both those spatial features of where oak trees are and how much pollen they're releasing, as well as the temporal dynamics caused by temperature differences across the city of Detroit. Now, the big question, though, is whether this model predicts airborne pollen at particular times and places. And I'm excited to say that when I compared this model of pollen release with airborne pollen from that data set that I collected with the pollen samplers, this model explained almost half of the variation in airborne pollen concentrations for oak trees. I want to emphasize that this is about 9 times better than simply using pollen measurements from the nearest pollen monitoring station as a proxy, which has been the standard practice in this field, at least as it applies to allergies. Allergists have long said that pollen is everywhere and you can't avoid it, but these results say otherwise. There are real pollen hotspots, and they are also predictable. So imagine having this extra resolution and accuracy in your pollen forecast. I think it offers up all sorts of possibilities, including ways for people to either avoid pollen hotspots or to take their medication ahead of time so it reaches full efficacy before they're exposed. These types of improvements could really improve quality of life for many people, uh, but for vulnerable people, including those with asthma or chronic obstructive pulmonary disorder, this is a really big deal. You may be wondering about the effect of wind and atmospheric dispersion on airborne pollen concentrations. And I can say it's very big and it should be included here. These models of pollen release will pair nicely with atmospheric dispersion modeling. Um, and I've collected some of the data needed to calibrate these models, but I haven't created them yet. Um, I also expect that the same framework could also be applied to fungal spores, which would have applications not just in allergy, but also in agricultural settings. So far, I've shown you how my framework works for trees, and in my next example, I'm going to turn towards ragweed, which is an early successional species that releases vast amounts of allergenic pollen. And to begin, I'd like to acknowledge Tiffany Carey, who is a mentee of mine starting in 2010 when we first started a pollen project together. And I'm really proud of the contributions that Tiffany made to this work. And she's now back in Detroit again, working with the National Wildlife Federation on environmental and social issues there. One of the things that Tiffany and I noticed pretty early on was that there was far more ragweed and vacant lots in Detroit. The city has demolished about 20,000 vacant structures since 2014, and here's a little chronosequence of what that looks like. So here's a building that is slated for demolition, and here's the exposed soil the next spring. By the fall, you can see little bits of green in the lot, and that is exactly the habitat where ragweed thrives. I quantified this habitat across the city of Detroit using remote sensing data and linked it with field surveys in order to create a map of pollen production across the city of Detroit, which I'm showing to you here. This approach worked pretty well, and I'd also think that similar methods could be used for understanding weed populations across agricultural landscapes. One of our findings was that some neighborhoods had lots and lots of ragweed and others had almost none. The neighborhoods that have many vacant lots and a lot of ragweed tend to be poorer. And in fact, you can just see the spatial correlation between ragweed pollen production and the percent of Detroiters living in poverty, which is shown in this bottom map. The people living in poorer neighborhoods also tend to have less access to health care and higher allergy and asthma rates. So I think this is an example where ragweed pollen exposure is actually an environmental justice issue too. I'd like to take a moment to talk about this statistic that I've just put on the screen, that Black people in Detroit are twice as likely to be hospitalized for asthma as white people in Detroit. Now, this isn't the type of statistic that we usually mention in plant ecology talks, but I think it's relevant here because interactions with plants could be making a certain amount of contribution to this situation. Luckily, in this particular case, there are some simple management recommendations for vacant lots. And in particular, it turns out that if you either mow regularly, the ragweed goes away, or if you leave the lot on its own, succession happens and ragweed is outcompeted. So I've now shown you examples of how I've quantified the ecological processes relating, relating to pollen release and how I've scaled this up to the municipal level for both oak trees and ragweed. But these are just predictions in a single city. And so the next thing to do is to work to scale this up to even larger areas. And that is what I'm doing as a postdoc at the University of Texas. My research here is on cedar fever, which is actually allergies caused by juniper pollen. Still, this is one of the most important allergenic pollen producing plants in the US, in part because of how much pollen it releases. Here's a branch of a juniper tree releasing pollen in January. A really big juniper tree of this species can release as many as 500,000,000,000 pollen grains each year. Anecdotally, we know that cedar fever has big effects on respiratory health in Texas, but that's never been quantified. Right now, I'm working on an epidemiological analysis to compare airborne pollen concentrations with emergency department visits for asthma. And the preliminary results from this study are showing that cedar fever is conservatively associated with about 5,000 extra emergency department visits each year. Since only the most extreme cases result in ED visits, the true effects of cedar fever on quality of life for Texans is astronomical. The way I'm going about working on this problem is to create similar models to what I did in Detroit. And to do so, I'm taking the same approach. I'm figuring out how much pollen individual trees produce, figuring out where these trees are and when they release pollen, and then using dispersion modeling. And the goal of this is to create near term forecasts and alerts for cedar fever sufferers in Texas. Now, to make this work at the state level, I've had to do something different because I can't drive all around the state to collect these measurements myself. So I started a citizen science project called Pollen Trackers through a collaboration with the National Phenology Network. Volunteers on this project are helping to collect those measurements of pollen release by junipers across Texas, and we are just about to go into our second field season next month. The goal of my work is to develop process-based models of airborne pollen at the continental scale. Models like this would fill gaps in our understanding of both gene flow and of how much pollen people are exposed to. Forecasts from this model would also be really useful for people to both reduce their own exposure and to better manage their medications. So there are a lot of potential benefits for having continental scale models of airborne pollen, and it would also help us to do things like understand the role of local pollen production versus long distance dispersal of pollen. This would have applications not just in the allergy world, but it would also be really important for understanding gene flow. Of course, creating a continental scale model is an ambitious goal, and so I've decided to break this up into several distinct research projects. The first of these projects is understanding where trees are and how much pollen they produce. This is a big task, but I think that it's feasible to do this. The way I propose approaching it is by combining data on what trees are where from the Forest Inventory and Analysis dataset, which now, in addition to including forested plots, it also includes data on urban trees. This dataset of tree locations and abundance could be combined with remote sensing data. And in particular, it could be combined with imagery from satellites like Landsat, as well as with LiDAR from JEDI, which is another potentially very useful resource for this project. These estimates of tree abundance could then be combined with allometric equations of pollen production. I have some ideas about how we could do a better job of making allometric equations faster and more efficiently. And I think a great place to parameterize some of those equations would be R0 forest. And I think it's also going to be important here to include some of the traditional plant ecology processes that are also going to affect pollen production. So things like competition. There are a bunch of applications for these data products, both for pollen, but also for understanding other ecosystem services and disservices, including things like, say, the spread of tree pests and pathogens across the US. I think that NASA would be a good potential funding source for this project, specifically through the Earth Science Research Program. In order to create continental scale models of pollen, we also need to create continental scale models of flowering time. To create this, I propose doing a few things. First, expanding my current citizen science project with the National Phenology Network, and they're excited about this possibility too. We could also get observations of flowering time from neon. Another possibility is to use some new pollen monitoring devices that I have been piloting here in Texas. And these devices, let me pull one out. These devices allow you to, it basically is a little Arduino controlled motor, which can be timed to spin a acrylic plate at a certain rate. And these can be placed underneath trees. And you can change the timing of this so you can get very fine resolution estimates of pollen release. So right now I'm piloting the idea of sending these devices out to people in my pollen trackers program so that they can both corroborate the data that they are collecting, because let's be real, citizen science is messy, but more importantly than that, they could augment it by having a finer temporal understanding of when pollen is released. These observations of flowering time could then be combined with granular climate data, and the combination of these things could be used to create flowering models. These could then be, these flowering models could potentially use close to real-time data sources in order to create near-term forecasts of flowering. Better and more accessible models of flowering time would be useful for many purposes besides allergies too. And let's see, so one example is the risk of frost damage to flowers. I worked a little bit on this project in graduate school, and I found one of the things that is clear in the literature is that climate change is increasing the risk of frost damage to flowers. And in fact, in Ithaca this year, there was snow on the ground on May 9th. So having a better sense of when things are flowering could be really useful for understanding and responding to frost risk. And this, I think, would be especially relevant in orchards. This project would likely be a good fit with NSF, and particularly with the Division of Environmental Biology that funded my graduate work. Ultimately, one of the major justifications of my research is to better understand how pollen affects human health so that we can do something about that. One of the projects I'm really excited about is combining high-resolution pollen exposure data with high-resolution asthma exacerbation data. The group that's doing this best right now is a company called Propeller Health, which uses smart inhalers that provide information on the time and location of each asthma inhaler usage. I've discussed combining our data with them, and we think there's a lot of potential to use this information, both the fine-grained pollen exposure estimates with the fine-grained asthma exacerbation data, to potentially create individualized dose response curves for people. This also opens up the possibility of personalized forecasts. And Propeller Health, this company, they already help users to manage their asthma. So there's a clear avenue for using these estimates of airborne pollen concentrations to change how people are managing allergic diseases, including asthma. I think a really good place to work on this project would be New York City, uh, in part because there are a lot of people with allergies there and a lot of good sources of data that I would need to create models of airborne pollen there, but also because I think there's a lot of potential to collaborate with physicians at Weill Cornell Medicine. Having these types of personalized forecasts and dose response curves would be a game changing project from an allergies perspective. And I think this would be very competitive for funding from the National Institute of Environmental Health Sciences. These research topics that I've been talking about, there's a multitude of little projects within them that would be excellent projects for graduate and undergraduate students. I look forward to working with students on this research in the future. And I also think that some of these topics uh, could apply well to both existing classes at SIPS and to new potential classes too, such as a class on the links between plants and human health. And since Chris mentioned it, this photo here is actually from the class she mentioned, this new Intro to Ecology field class that had about 100 master's students in it. In that class, we used active and inquiry-based pedagogy, and those are approaches that I hope to use in teaching in the future as well. To end my talk, I have two conclusions. First, that process-based approaches offer an effective framework for modeling airborne pollen. My second conclusion is that the combination of these research themes are a very useful toolkit. And today I've told you about how I've applied them in a few ways in my own study system, but I'm also excited to see how my methods could be used to answer questions in other systems, including farms and orchards. So over the next couple of days, I'm really excited to chat with you all about ideas like this and also about the potential for collaboration in the future. So, with that, I'd like to acknowledge these institutions for their financial support. And I'd also like to thank some of the many people who contributed to this research, both at the University of Michigan and now at the University of Texas at Austin. So, with that, I would be happy to take your questions. Thank you.
All right. Thank you, Dan. Really appreciated that. Are there questions for Dan? You're welcome to unmute or put them in the chat, or I'm trying to open up all my windows here. Raise your hand. We have so many options. Nina, did you have a question?
Yeah. Hi, Dan. How you doing? Thanks for the presentation. I just wondered about, obviously pollen does cause allergies and cause reactions, but, is there a possibility of looking at this as a control group in a forested area as opposed to an urban tree area? I would imagine there'd be more trees in a more forested area than one in an urban area. And I just wondered if you looked at sort of a control group of trees.
Yeah, I think that's a really interesting point. Um, I think there's a couple of reasons why I've focused in on urban trees, specifically that we interact the most with the trees that are closest to us, and and there's also the incremental effect of an individual tree in a city is much stronger on health than that of a tree in a forest. I think there are a bunch of really cool questions that we can answer using these same approaches for more rural forests too, though. And I think though, one of the more interesting sets of questions is about gene flow. But I think the same approaches could be used for that. When it comes to allergies, there's this thing where people who live in rural areas also tend to develop fewer allergies compared to people living in cities. So that is another reason to focus more on urban areas.
Actually, that's another issue, that there are other aspects of urban life that cause allergies than pollen. Basically automobile exhaust, hydrocarbons. I mean, that's a big factor, and that's not really factored in with your model. And I wondered if you had some response to that.
Yeah, certainly. That's a great question. And so it's not factored into my model of pollen exposure, but it is factored into epidemiological analyses. And so that's where we need to do things like figure out the effect of various air pollutants, including particulate matter, for example, and ozone, which interacts a little bit with pollen. To And that's also where we need to do things like understand the effects of, say, viruses on emergency department visits, just to reference the epidemiological analysis that I'm doing now. So all of those things are important and will affect the proportion of some health outcome that is attributable to pollen specifically.
Because trees also reduce particulates in areas. I mean, they do, particulates do land on leaves, and so the particulates in the air tend to be less where there are trees. So there's a lot of different competing issues here, and I wonder if pollen alone is the major issue in terms of asthma.
Yeah. Um, I appreciate the pushback here, and I think it's what you're making are very good points. And however, just because there are other issues doesn't mean we shouldn't understand allergenic pollen. And moreover, these same approaches are really useful for doing things like understanding how much particulate matter these trees can filter out. So I think that these are great points, and I'd be happy to go through it more with you, because I think pollen is just one issue, and ultimately we need to weigh both the ecosystem services and the ecosystem disservices that these trees are providing. And that is ultimately what should govern our decisions about tree selection.
Joss, you had your hand raised?
Thanks for a great talk, Dan. So in the beginning of your presentation, you mentioned that, I think you said pollen was becoming more allergenic. Did you mean on a per pollen grain basis, or contextually in the context of other environmental components?
Well, I'm afraid it's both, unfortunately. Yeah, so we have some good lab experiments that show that plants grown in higher carbon dioxide concentrations produce pollen grains that are, on a per pollen grain unit, more allergenic. And so a lot of that work has been done on ragweed, and yeah, it's on a per pollen grain basis.
So to kind of continue that line of thought, I guess many secreted cell wall proteins in pollen are allergens. And is anybody looking at that, looking at the allergenic profiles of cell wall protein clusters?
Yeah, I think there's a little bit of work being done on that. I would have to look in uh, my literature folder and see exactly what is being done there. It's certainly something which is of interest to a lot of people, and yeah, so I'll look a little bit more at my papers and then give you a better answer in our one-on-one meeting.
Yeah, we can talk later. Thanks.
There's a question in chat on pollen morphology.
Yeah, that was me. Yeah, I really liked your talk, and I really want to say that I like the use of Arduino to measure pollen release. I think that's really cool. But I had a question about whether or not you're incorporating pollen morphology to inform like your flowering models. And the only reason I asked is because I'm not 100% sure about pollen grains in oak across oak species. I would go out on a limb here and like ask you, do you know if like they have like different morphologies across species of oak, and maybe if that's something that can affect like whether or not they're allergenic?
Yeah. Oh, that's an excellent question. And so I have a couple of responses. First of all, oak pollen grains generally look fairly similar. And so the standard practice in the field of aerobiology is not to even try and identify what pollen grain comes from red oak versus from white oak or something like that, because you can't do it reliably. That being said, there are differences in, say, the allergenicity of different closely related species. And so even if it's not something that we can just see morphologically, just using a microscope, it can have important consequences for allergies. Does that answer your question?
So when it kind of gets at it, like when you mean like, I guess, characteristics of allergenicity, are you referring to like the shape of like the pollen grain?
I'm referring more to what's inside the pollen grain. And a
Okay.
lot of them, and so there are a variety of proteins inside of pollen grains and substances which trigger allergic reactions. And so those can vary a little bit. It is easier to quantify the variation in the cellular content, and specifically in the allergic reactions that the contents of a pollen grain produce, than it is to say that, hey, like there's a slightly like thicker exine on this pollen grain than that one, something like that. So it's more about the protein composition than it is about the shape.
Okay.
However, shape can be important for things like how easily a pollen grain bursts and releases all
Right.
of that stuff into the air, which is a really important part of this whole story that I haven't had a chance to talk about today.
Yeah, I was just kind of thinking about if, like, for instance, like the exine is thicker on one pollen grain versus like the rest of the lot, then it would potentially disperse differently.
Yeah, entirely. And that, I think, is something that we'll need to do a little bit more of taking into account when it comes to dispersion modeling. And things like the amount of water that a pollen grain picks up is pretty important there. Yeah, so that's a great point.
Cool. Thanks. I really enjoyed your talk.
Thank you.
There are a bunch of questions here from a fellow plant pathologist. Interactions between pollen and fungal spores, as many fungal spores are also allergenic. Do your models take into account, or yeah?
Yeah, totally. And I think so. You may have been a little surprised by how little we know about airborne pollen. You would be even more surprised by how little the allergy community knows about airborne fungal spores. They are smaller, they're much harder to identify just with a microscope, and so we know a lot less about that. That being said, I think that there might be some possibilities for using the framework that I've done here, like figuring out source strength and the number of particulates released. I think there might be some potential to do that with fungal spores too. Um, and I should also mention that, yeah, fungal spores are really important to allergies too. They generally are. There's a few National Allergy Bureau stations that measure them, that measure fungal spores, but it's not as common to include that in this type of analysis.
And David Rossiter asks if you noticed differential wind effects in the Detroit study. He noticed cooler temperatures just inland from Lake St. Clair, and could that be a wind effect?
Yeah, I've wondered a lot about the effects of wind there myself. Um, I think that it's likely that some of the distance from water is affecting things like temperature. But that being said, if things like wind are uh, something of can are adding extra variation. Once we include that in our models, then these models are just going to improve. And so I think that's an area which I would love to add more in on, because it's just going to allow me to make better predictions of airborne pollen once we do things like incorporate variation in wind across the city, as well as, like which direction the wind is blowing and how strong and all that stuff too.
And somewhat related, Chelsea asks, can you use your methods to track pollen for plants that are not wind pollinated?
Oh, yeah, it's a really fun question. So, right now, my co advisor is Shalini Jha, who, and all of her work is on insect pollinated plants. And so, I think one of the really cool topics here is, could we use some of these same methods to figure out um, uh, the amount of pollen production across like an agricultural landscape or a wild landscape? And actually, a buddy of mine from grad school, Aaron Iverson, who just has been at Cornell for the last few years and now got a faculty position elsewhere, we've been bouncing around the idea of using some of these same methods to get a sense of what the foraging landscape would be like for a pollinator. So lots of cool possibilities for that.
There's one other question, which again, as a plant pathologist and someone who I have a project on rust fungi. So the spores of rust fungi can be highly ornamented. And the question is actually, can your models be applied to rust pathogen spores? Because several people online today are allergic to rust pathogen spores. And I think to some extent you already spoke to that, but they are, the spores tend to be wildly wind blown.
Totally. Well, hey, I say let's give it a shot. Let's see what happens.
And one of our hemp students says, can't wait to see this type of modeling applied to pollen and pathogens in the hemp industry. Any final questions for Dan before we let him go off to his next meetings? Chelsea?
I just had a quick one, and it kind of goes back to your Detroit study. So I apologize if I'm getting some of the information wrong, but I was thinking about, you were mentioning that that there's a preponderance of cases where there's an increase in certain communities for being hospitalized for asthma in particular areas that are also associated with having greater densities of ragweed. Did I get that right?
Yeah, there's a correlation there.
Okay.
The causal links are a little more tenuous, and I'm not going to say that, and I cannot in any way say that it is ragweed that completely causes that, but there could be a fraction of that which ragweed contributes to.
So I was thinking about that from like the public health. I mean, is this a place where the medical practitioners could go in and say, hey, look, this is an area that doesn't have a lot of asthma knowledge? Because I can imagine also certain communities just aren't even exposed to the healthcare system that would provide them with understanding of how asthma can be both provoked and prevented in children. So just thinking if this could be a way that a collaboration with the medical community to say, hey, listen, these are areas that are potentially high risk. These are communities where you could go and do some public health educational services or offer clinics or things like that.
Yeah, I think that is one of the most important potential applications of this work. And my other co-advisor here is an allergist, Elizabeth Matsui, and her claim to fame is doing studies in Baltimore that looked at how differences in pest control, specifically with mice, affected people's risk of developing asthma. And so I'm a plant scientist, but I think there's a lot of room here for these approaches to be used for public health interventions. And that's why I'm currently, as a plant scientist, based out of a medical school here. So I completely agree. Thank you for the softball question. I love it.
And Bill Crepay notes that the methods could be used in forensic botany. And as an aside, Bill teaches a class, oddly, in forensic botany.
Yeah, completely. There's actually this really interesting project that's being done largely at UT Austin now, that's funded through the Murray mechanism. This is the Department of Defense, and they have this it's this massively funded project to use some of this to use pollen as a tracker for figuring out where things have been. So yeah, totally super useful for all of that as well.
And Carlin has a question, but she's going to ask it tomorrow because if I don't stop this, people will riot. It's been great. Sorry, Carlin, I love you. It's been really fun talking with you today, Dan, and I hope you enjoy your conversations with folks across the School of Integrative Plant Science. So everyone, please join me in thanking Dan for a great seminar.
Thank you. And I'll look forward to chatting with you all over the next few days. Looking forward to it.
Cheers.
This has been a production of Cornell University, on the web at cornell.edu.